Hi, everyone. Thank you for joining. We're going to get started at about 2 past the hour. Thank you all. Okay, I have 2 past on my computer, so let's go ahead and get started to give our speaker enough time. Thanks everyone for joining us today for our HESI Eco Risk webinar. I'm going to go through a few slides just with background and logistics before I turn over to our speaker. So this webinar is hosted as part of our HESI's Next Generation Ecological Risk Assessment Committee. I won't read the whole mission, but just to say this committee brings together people from academia, industry, government, and other research institutes to support ecological risk assessment with a focus on alternative non-animal testing methods. For the webinar today, we will record it and make it available to registrants. Other than the speakers and panelists, folks cannot speak, so they can type their questions in the question box. I'll show what that looks like on my next slide. If you do have any trouble today, please give me an email. My email is listed there or on our website. And this is what it looks like to ask a question. There's 2 different views that should look something like this if you're on the desktop app or on the web. So you can ask your questions there and we'll have time at the end. Just to say for our eco-risk portfolio, we do have many projects going on. I'm happy to share these slides with folks, and if anyone's interested in working on a particular project they're not a part of, please get in touch with me or other HESI staff. So with that, I'm really excited to introduce our speaker today, Dr. Jochen. Dr. Jochen studied environmental sciences and ecotoxicology at the University of Koblenz-Landau. Forgive me, I'm not German if I'm mispronouncing anything, and that is based in Germany. There, he earned his PhD working on the impact of pesticides in stream ecosystems. He continued there as a postdoc, working as a research team leader and teaching, among other things, environmental statistics. After further education in data science, Jochen then started his own business where he applies data science tools such as machine learning techniques to environment-related problems. We're really excited to have you. Let me stop sharing my screen and I'll turn over to you. So you're now the presenter and should be able to share yours.
Does that work?
I see your screen and it's going, yep,
Perfect.
that looks perfect.
Cool. Yeah, thanks Connie for the kind introduction and the opportunity to present some of our ongoing research here today, for which I worked obviously with some colleagues, particularly to name here are David Dreyer, Nika Garlick, and Maxime Bourgeot. Ecotoxicity or toxicity data sets are by nature sparse. That means we do not have information of all possible combinations of chemicals and species. And if we search for some data, maybe on some database, we might come up with a situation like this. And I'm not sure if you can see my pointer. If not, maybe Connie can give me a sign.
We can. Looks good.
Yeah, perfect. So we have a combination of 2 category variables, like a chemical identifier, a chemical name, and a species identifier if we are searching for information on several species, also category variable, and then we are usually interested in some kind, or maybe interested in some kind of effect value, like an LC50 or something. And because these data sets are usually sparse, we find some data gaps here. And these are two different situations here. Like for chemical 3, there is only one information for one species missing, which would not be a problem actually if we had more species. But already the second, here, these data gaps for chemical 4, we see we don't have any information available. But this is not a big problem, so we can use just frequently used approaches for modeling and predicting this information. This is a very typical situation that we might find for QSAR modeling or QSTR modeling, so quantitative structure activity relationships or quantitative structure toxicity relationships. And the basic idea how we get to that prediction is we first substitute the categorical chemical name species by some kind of computer readable representation of that chemical, and from that we can use other tools to estimate some kind of numeric variables, chemical descriptors or fingerprints. And now with these numerical variables available, we actually are capable of predicting both of these situations. So for the rest of the talk, I will call that cross-chemical predictions. But now extend that problem a bit. We are also interested in toxicity values or other effect values for a third species for which there is no information at all. Now we have a problem because this species variable, the species descriptor is still a categorical variable. And so we are not capable of doing any predictions beyond the levels of that categorical variable that was present in our training data. And so the basic idea we came up with was that we should also try to replace that categorical species variable by some kind of numerical descriptors. And our idea was to use physiology for that, because we know from a lot of studies that physiology is an important driver of species sensitivity. And so in theory, this might look like this. We have chemical descriptors. Now we also have physiological descriptors, numerical variables, and we should be able to also predict these last missing data values. So do both cross-chemical and cross-species predictions. So, but now the next question was where to get that biological information from, what kind of physiological information should we use? And for our ongoing research, we started using model parameters from so-called or which are coming from so-called dynamic energy budget theory. This is a theory describing life history of individual organisms through a bioenergetic lens and was shown to adequately describe physiological processes of energy acquisition and allocation within organisms. Models based on this theory use a consistent mathematical formalism, so species and their physiology can actually be compared based on that parameters. And there is a lot of data available regarding depth parameters, for instance, in the well-curated AtMyPet database. The most basic depth model, which is called the standard model, assumes that energy is assimilated by an organism with a surface area specific maximum assimilation rate called PAM into reserve. From that reserve, there is an energy flux with a specific energy conductance rate V. And this energy flux is then fractioned, and the fraction kappa of that energy flux goes into the soma, where the energy is first used to for maintenance purposes, so protein turnover and so on, and we have a volume-specific somatic maintenance rate called PM. And the rest of this energy fraction is then used for growth. And then we have a volume-specific cost for structure and a parameter called EG. And then the base model or standard model assumes 3 different life stages of an organism. That is egg or embryo, second stage is juvenile, and the last one adult. And for the transitions between these stages, we have threshold parameters, one that is a threshold for feeding, so birth, and one threshold for reproduction, so puberty, EHB, EHP. And when that modeling is getting a bit more complicated, then we have also other parameters involved that can be classified into primary. So these parameters have a close conceptual link to the underlying physiological processes. We have compound parameters which can be calculated from the primary parameters, and we also have life history parameters available. And you can see here this list of 9 primary, 3 compound, and 3 life history parameters, and all of these parameters we tested in our modeling approach. So we wanted to provide a proof of concept that this idea is actually working. We restricted our exercise to pesticide toxicity in freshwater fish and invertebrates. Pesticides, as they are a well-researched and regulatory important group of chemicals, freshwater because we have a common exposure unit, and fish and invertebrates also because they are taxonomic groups that are of regulatory significance. And we wanted to answer if the involvement of that parameters in QSAR-like approaches allows for reliable cross-chemical and cross-species predictions. And we were recently asked not to call our approach a QSAR because technically QSARs don't involve any biological information. So we came up with a new name, but just for the sake of simplicity, when I talk today about models, QSAR-like approaches, QSARs, this also includes our approach. Additionally to answering this basic research question, we also wanted to make our models flexible, accessible, and explainable, and I will talk a bit about what that means later in the presentation. We made use of a range of different data sources to create our data sets. First of all was the EnviroTox database, where we took effect values, water solubility, and log KOW from for representing fish toxicity. We chose 96-hour LC50s, and for invertebrates, we chose 48-hour LC50, EC50 values. Not to create any confusion here, so EC50 values were all based on immobility, so as a surrogate for mortality. And as I said before, only freshwater species were selected here. For our chemical fingerprinting, we chose 2 different approaches, or tested 2 different approaches. First, ToxPrints, which is, for instance, also recommended for QSAR modeling by the FDA. And the second one was a newer approach, moderate, which contains a lot more, or provides a lot more of these chemical descriptors. That parameters came from the AdMyPet database. And as I said before, we tested 9 primary, 3 compound, and 3 life history parameters. To be able to classify our chemicals, or the chemicals that we took from the EnviroTox database, into pesticides and non-pesticides, we made use of the lists provided by the resistance action committees for fungicides, herbicides, and insecticides. And we also took some additional physical chemical parameters from the ComTox chemicals dashboard. These are our data sets with which we came up. You can see on the top the data sets for fish and invertebrates. As mentioned in our title, we used a machine learning approach. There we usually split our data sets in a training data set and a test data set. Testing is usually, as we also did, 20% of the total of the data. So you can see both data sets here for both taxonomic groups. You can see we had a lot more samples available for our fish models than we had for invertebrates, and the same for the number of pesticides and the number of species. So for fish, it was in total 92 species, for invertebrates only 20. And when we had a look into our data, we also found that species representation was highly imbalanced, which makes sense because they include standard test organisms like the rainbow trout or the water flea Daphnia magna, which are very well represented in our data sets, but we also have species that occur only once. And this will become a bit more important in a second because we try to counterbalance this phenomenon. As our main algorithm for modeling, we chose the machine learning algorithm random forest, which is based on a technique that is called bagging. Bagging is short for bootstrap aggregation. Where is my pointer? Here it is. So what we do is we have our training data set and we take repeatedly with replacement, that's where the bootstrap is coming from, subsamples of our training data set. And on these subsamples, we train so-called weak learners. In our case, for random forest, these are simple decision trees. And finally, when we make predictions, we just aggregate all these weak learners to get a strong learner, and that's a random forest then. So it's a tree-based ensemble learning method combining robustness against overfitting with a high degree of interpretability, which is important for our goal to have explainable models finally. And random forest were also shown to outperform other approaches when used for toxicity prediction. We used random forest implementations and also the general machine learning framework in R for our study. And before we built our final models, we made a preliminary modeling step that was thought to reduce dimensionality of our data sets. You have seen the number of descriptors that were available. So these are a lot of variables in our data sets. So we tried to reduce that before we went into final modeling using a technique called recursive feature extraction. So we make a modeling step that we find, okay, find out about the importance of the different variables or features, and we just cut away the 10% least important. Repeat that, cut away another 10% down to 40% of the original number, and we test that by a 5-fold cross-validation. And with that optimal set of features, we then go into our final modeling step, where we also performed a grid search for hyperparameter tuning with 10-fold cross-validation. And with that basic approach, we built a lot of different models. First of all, what we call the baseline model as a reference. This only included ToxPrints as chemical descriptors, the categorical species variable, and the two base physicochemical parameters, water solubility and log KOW. And then we did a lot of changes to the data set underlying that baseline model and also changed our modeling a bit. First of all, we changed feature composition. So we replaced the categorical species feature by depth parameters, tested all possible combinations of the 9 primary parameters, all possible combinations of the 3 compound parameters, and also all combinations of the life history parameters. The next change we applied was replacing ToxPrint chemical descriptors by moderate. And finally, we added additional physicochemical parameters to the data set for our model. Then we did some changes to the data set regarding plausibility. If we go to any ecotoxicity database, there's a lot of effect values that are actually much higher than the reported water solubility. And this was also for us the case. And so as this relationship between water solubility and effect value is so important for many models, we decided also to try a data set that was restricted to only plausible combinations of these two features or these two variables. And the last changes we applied were related to balancing our data sets. And the splits we usually do for machine learning into training and testing, they are done randomly, which can come with problems. So we also tried a semi-random approach that made sure that the distribution of our targets or our effect values were, we're more or less equal in both training and test data set. And the second balancing strategy was related to what I've shown you before, to this imbalance in species representation. So we added some weighing approach to our modeling pipeline. And in a first step, we applied all of these changes individually for both taxonomic groups, resulting in more than 1,000 models that we built. And with the results of the first step, we then went into a second one. So all of the changes that resulted in better models than the baseline model, we combined factorially, resulting in another approximately 500 models. From the second step, we then selected two final models, one for fish and one for invertebrates, and tested them against alternative approaches to make sure that our approach, that our choice of machine learning algorithm, model pipeline, and so on was actually suitable for the task at hand. So we tested two different machine learning algorithms. First one was support vector machines. This works completely different than random forests. So what we want to find is like a mathematical description for a hyperplane that best describes our data. There are different ways of performing support vector machines called kernels. We tested two of them, most commonly used linear and radial kernels, and also support vector machines were already shown in the past that they can result in high predictive power for QSARs. And the second algorithm that we used is a very novel one called local cascade ensemble, which is thought to combine the strength of bagging, which I showed you for random forests, and boosting. So what is boosting? Again, we take our, we take our training data set. This time we don't take a subset, but just train a so-called weak learner again. And now we take the residuals of that model and build another weak learner on that. Then we take the residuals of that model, build another weak learner on that, and so on. And finally, again, we aggregate the predictions of all the weak learners to get to a strong learner. And then we also tested an alternative to our preparatory modeling step. You might remember that we took recursive feature extraction for that in our pipeline. And we tested that against principal component analysis, another very commonly used approach for reducing dimensionality. After this step, I told you before, we were highly interested in getting to explainable models finally, so to assess feature importance for our predictions. But with the data sets we used up until then, there was a problem, which is called multicollinearity. Just imagine we have these hundreds and hundreds of features in our data sets, and a lot of them are actually highly correlated with each other. And so when we are interested in feature importance finally, so these importances or these highly correlated features, they just dilute their importances. So we cannot really tell how important they are. And so we need to correct for multicollinearity to do so. And so we subjected our data sets for the final or selected models to an algorithmic correction for multicollinearity, and then repeated our modeling, so both the recursive feature extraction step and the random forest step. And then we had these corrected data sets or corrected models on which we really were able to assess for feature importance, and we took again a novel approach for that from the field of explainable machine learning or artificial intelligence, which is based on threat values, shapely additive explanations with fundaments in cooperative game theory. So basically what you do or what you're interested in is the outcome of a game in any combination where a certain player is present or absent, and translated to our modeling situation. That means we are interested in the predictions we get for any combination where a certain feature or independent variable is present or absent. And again, we made use of an R package implementation for getting these values. Okay, so let's have a look at some results. I'll guide you through these tables. For reference, here in the red rectangle, we can see the results for the baseline model for fish of the first modeling step now. So the model having ToxPrints as chemical descriptors, involving the categorical species name variable, and only the two base physical-chemical parameters, water solubility and log KOW. The most important column now to look at is this one. This is the R squared we get on our test data sets, so our validation data sets. And we can see that already for the base model, we get a decent R square, around 75%, 76%. But also that almost all changes that we applied to that combination resulted in higher predictive power, up to around 85, 86%. So we can see that the involvement of most depth parameter combinations actually improved predictive power. The replacement of ToxPrints by Mordred improved predictive power, and that was already shown before in other studies. The semi-random splitting approach, the additional physical-chemical parameters, but the best performing model actually was the one that we restricted our data to plausible combinations of the effect values and water solubility. And this makes sense because water solubility is such an important predictor for effects, and also was already shown to be very important for fish toxicity before, so that it makes sense that we need to have some plausible relationship between these two variables. Let's go over to the results for invertebrates during the first modeling step. Again, as a reference, the baseline model, we can already see, okay, this was the worst performing model for invertebrates. Again, here we have our test R squares, and yeah, with the baseline model here at the bottom, it's very clear that all changes that we actually applied improved our model performance, but particularly did so when we either involved our balancing strategy for species representation, so involving weights, or replaced the categorical variable species name by depth parameters. And the question now was, why is it so much more important to have these changes for invertebrates compared to the fish model? We know that there is a correlation between species sensitivity and phylogenetic similarity, and the invertebrate training data set featured a much wider taxonomic spread of species. Fish only separated at the order level, while invertebrates already separated at the phylum level into arthropods, mollusks, and rotifers. And as physiological dissimilarity increases with taxonomic distance, approaches either tackling imbalances in species representation or replacing that categorical feature species name, which is just a name tag for the model, by features containing meaningful physiological information, become naturally more important in data sets with wider taxonomic spread. Sorry, I needed to put that little go-to screen aside. So now we're back in full presenting mode. Okay, so and with these changes that we found in step 1 that were promising, so they improved our model performance compared to the baseline models, we combined them factorially for the second step. And here you can see the results of the 10 best performing models for both fish at the top here and invertebrates here. And again, here our test R square column. So what directly comes into the eye, again, for fish, we achieved only in quotes around 85% R square. So we didn't achieve a further improvement here for fish in the second step. But now what we can see is that all the best performing models had depth parameters involved instead of the species name variable. And for invertebrates, we can see that we had a further improvement in model quality and predictive power. And so now both fish and invertebrate models were of comparable predictive power. And yeah, we saw that we indeed could successfully replace the categorical species variable by depth parameters. And from these final models, we chose two, one for fish, one for invertebrates, based on number of features, which was supposed to be as low as possible, the predictive power of the models, but also the involvement of certain depth parameters about which we had previous knowledge from literature. And with these two models, we then went into the next step and tested for the suitability of our modeling approach, our modeling pipeline. And here you can see the results for our selected random forest model compared to the SVM approaches, the local cascade ensemble, and also the random forest that were built after reducing dimensionality using principal component analysis. Here again, the test R square, and we can see that all of these approaches actually for fish resulted in comparable predictive power. For support vector machines, it was shown before that they can result in similar predictive powers for toxicity prediction. Yeah, but for invertebrates, the picture looked a bit different. Here we can see that only local cascade ensemble, so another tree-based algorithm, achieved a comparable power than our approach. And yeah, for SVMs, that was like that bad quality of that models was a bit surprising to us because there are other invertebrate QSAR approaches out there that showed the potential for much higher predictive power. But again, we believe that this is mainly because of the wide taxonomic spread in our data set. So these previous models that we found in the literature were mostly based on smaller subsets of this animal group, like crustaceans or subgroups of crustaceans. And there is another problem that might be involved here. It's called big P little n. Big P stands for we have a lot of features in our models with a comparably low number of samples involved. So we can see for the two support vector machines here, 400 and approximately 1,000 compared to only roughly 300 samples, and SVMs were shown before to be in certain situations susceptible to that problem. Okay, so we believe that our modeling approach was suitable for the task at hand. None of the alternative approaches resulted in better models, and so we went on into the next step where we wanted to assess feature importance in our models. We first applied the algorithmic correction for multicollinearity to the data sets that resulted in our selected models. In both cases, for fish and invertebrates, dropping approximately 850 features, but still compared to conventionally used strategies for multicollinearity correction, we spared approximately 5% of features which might have contained in important information for our modeling. And with these corrected data sets, we then repeated our modeling, so both the recursive feature extraction and the random forest step, and found that our final dimensionality was even lower than when using PCA. You can see the numbers of the number of features for PCA approach here in the brackets compared to this here. And also we had comparable predictive power in these corrected models to the uncorrected counterparts. Uncorrected models, you can see the test R squared here and the test R squared for the corrected models down here in the table. And the big pro now was that our models were actually fully explainable. And with that, we actually were able to apply our SHAP value calculations on which we based our feature importance assessment. And this is the results for our fish model, the 20 most important features in this model. And non-surprisingly, for fish, we can see that water solubility and log KOW came out as most important. But we can also see that besides different chemical descriptors, also all 6 depth parameters that were involved in this model are among the top 20 most important. For invertebrates, the picture again looked very different. Here, as the most important feature, one of the depth parameters came out, e.g. You might remember from the very beginning of the presentation that this is related to the costs of growth, and our model associated animals, species with a high cost of growth, so high EG values with low toxicity values, so high toxicity. And this might be explained because high costs of growth usually are associated with a slower growth of the species, so there's less potential for dilution by growth. So by growing, animals can dilute the internal concentration of a toxicant, resulting in less damage at receptors and lower toxicity. And also pretty important here, the parameter PM, so the cost of maintenance for a species. And that was also repeatedly shown in previous studies that high costs of maintenance are associated with higher toxicity. Again, all 4 depth parameters that were involved in the invertebrate model were also among the top 20 variables. Okay, so we actually successfully replaced the categorical species variable by numeric physiological descriptors. We built flexible, accessible, and explainable models with our approach, but still there are obviously some limitations with our models, and I just want to briefly talk about one of them, the so-called applicability domain. I told you in the very beginning that ecotoxicity data sets are by nature sparse. What does that mean in numbers? Of all the possible pairs of the species involved and the chemicals involved in our data sets, only 5 and roughly 10% of them were covered by our training data for fish and invertebrates respectively. That means in turn that not all regions in the feature space are represented equally well. So what is the feature space? So there's just a multivariate space of all the feature value combinations that we had in our data set. And to go a bit deeper, for some features, independent variables, that meant that we found correlations, relatively high correlations between the actual feature values and the absolute model residuals. And the direction of these relationships was mostly determined by the regions of lowest density of that feature. That means if we have a feature combination that is not well covered by our training data, it is relatively likely that the prediction we make on that is not very reliable. What can we do about that problem? We need to further extend our training data set. We already work on that and have some ideas, and we're able to increase sample size more than tenfold. And so this looks very promising. And finally, when we then are happy or could not further improve the coverage of regions in our feature space, then we need to carefully characterize that applicability domain to make sure that our models are not used for predictions, for feature combinations where it's not well trained for. Despite these limitations, to our knowledge, what I showed you today is the first machine learning-based diffuser-like approach for ecotoxicity prediction that is capable of flexible cross-chemical and cross-species predictions beyond the levels that are present in the training data. The predictive power of our models compares favorably with existing models and tools for acute aquatic toxicity prediction, and it is applicable with a minimum of available information. So finally, you only need the name of your chemical, or even better, the computer readable representation of that, and the name of the species you're interested in, and you can go to databases or use tools that are freely available to estimate all the all the features, all the independent variables that are needed as input for our models. So they are highly flexible and accessible for a number of applications. And just to name a few here, we could use them to supplement risk assessment by including legally or practically non-testable species. We could use them to scrutinize community-based risk assessment approaches, such as species sensitivity distributions regarding their productivity for the mentioned species, and also potentially in future we could use them to replace some animal testing. And one point that I cannot stress enough is that our models are indeed explainable, and we already have seen a global analysis of feature importance for our models, but we can go even deeper, because these Shep value calculations, for instance, they are based on local predictions, and we also can use them for new local predictions, so single point predictions, and find out how did we get to these predictions, how important were the different features for that. And just to give you a visual example how that could help us, we have here Shep value decomposition for the minimum prediction on the left-hand side from our fish test data set, and on the right-hand side for the maximum prediction for the same data set, so over here. And not really surprising, we can see that most important features for these two predictions were also the ones that were most important on the global levels, on the whole model level. But there's more important and interesting information. Here we can see that for the minimum prediction on the left-hand side, there are some depth parameters involved. So that means that in this situation, we actually tested a relatively sensitive or susceptible species. And there is also interesting information provided by the chemical descriptors, which are not in the same order as on a global level. So for instance, here's third most important feature we have, descriptor called ATS-C2D. This is an indicator of molecular flexibility by single bonds. We have a value of approximately -3.4. And here we can use our feature dependence plot based on Shep values. We can see -3.4 is a relatively low number. So we are talking about a relatively rigid molecule here. And our model associates such rigid molecules with lower effect values, so with higher toxicity. And on the other hand, we have here as an example eta d epsilon d, which represents the contribution of hydrogen bond donor atoms. Again, a feature dependence plot here, and we can see that it has a 0.11 as a value, which is relatively high, and high values here are associated by our model with high effect values, so low toxicity. And this kind of information might come in quite useful, for instance, for research and development of pesticides, but potentially also other chemicals, because it might help us to better understand and get new insights into molecular level drivers of toxicity, and thus potentially also support future molecule design and the development towards green chemistry and toxicology. Just to briefly wrap all that up, we wanted to know if the involvement of depth parameters in QSAS allows for reliable cross-chemical and cross-species predictions, and we believe we were able to show that, and we wanted to combine that with creating flexible, accessible, and explainable models. And we also think that we were able to do that, but we are well aware that there is still a lot of work to do until these models could potentially be used in practice. Thank you for your attention.
All right, thank you so much for giving that talk. Really interesting. We already have a few questions in the chat. If others have questions, please go ahead and put it in the question box. So I'll start reading these and we'll go through them. There's a couple of questions from Richard Robinson. The first is, why would depth parameters be relevant for species-specific isoforms relating to MIE for apical toxic effects? Or are the effects considered in the proof of concept not highly related to such specific interactions? So that was a mouthful for me. I think it has to do with relating these to an AOP and an MIE. That's my understanding of the question, but we could also get clarity.
Yeah, maybe, or maybe just explain the abbreviations to me.
Oh, sure. So MIE is a molecular initiating event. If you're working on an AOP framework, that's what's on the left side. So, you know, the chemical interaction that then initiates the rest of the steps.
Yeah. Okay, so the question is how that is related to putting depth parameters as input into our models, right? So that I understand.
That's my understanding. If we're getting that wrong, Richard, please write in and maybe clarify.
Yeah, so I just answered the question the way I understood it. So I mean, the basic thing I think we did here was to provide our models with two sides of information. Yeah. So what is usually done is we get a lot of information about the chemicals. That's also what we did. Yeah. So instead of giving them just a name, we provide useful information that is actually somehow related to toxicity, whichever way. Yeah. So about, is it more water soluble? Is it not water soluble? You can go into that compartment, stays there, whatever. And I think the big pro is now that we also started to do that in a comparable fashion on the species side, and provided some information, maybe not directly about receptors or the places where toxicity is happening, if I understand the question correctly, but about processes that are directly related to how strong toxic effects can be in an organism. Yeah. So I mean, it's well known that all these processes regarding energy allocation with a species are highly informative about the susceptibility of a species, of an organism, for toxicity, yeah, towards chemicals. Not sure if that answers the question.
I think that's a good start, and maybe it's a thought on, you know, are these techniques sort of agnostic of the known chemistry, or do they build on it? And maybe that's a thought for how we use these data, like you mentioned in risk assessment. Okay, thanks for that, Richard. If there was nothing more specific for that one, please feel free to write in. Richard also had a question related to model selection. So did you perform model selection, tuning on subsets of the training set? He also then said maybe this was not so relevant if you use default random forest hyperparameters without feature selection. Would you consider nested cross-validation? So I think it's just kind of asking about your model selection and training set there.
Yeah. So I think it's like a combination, right? It's feature selection and model selection, how that was done. Feature selection was based on the recursive feature extraction technique. Yeah, so that was combined with random forests, and so we had that algorithm running for finding our optimal subsets of features. And then, yeah, model selection was purely based on our validation data sets, which were comparable for all the models that we tested, and mostly based on random splits, as I told before. Yeah, and yeah, so based on the RMSE and R squares that we gained on our validation data sets.
Great, thank you. And forgive me if I'm saying this name incorrectly. Yeon Sion asked, are the results for page 23 published?
So
For slide 20.
none of this, yeah, okay. So none of this is published yet. So we just received some minor revisions on a paper for that. So we hope that during the next days or weeks we get that published. Yeah.
Great, that's exciting. Okay, another question from Richard. Do you think the applicability of your models could be assessed purely upon the distance of the model or the density in the depths plus descriptor feature space for the training set? Or might extrapolation to very different species not be appropriately captured by DEB?
That's true. So both, I think, are important. The latter we did not do yet because our training data sets were relatively small, and or our data sets in general were relatively small. But now we are working on extending them, and I think I mentioned that we already have like 10 times more data now available. And now, as one of the next steps, and also like for assessing applicability, yeah, true quality of really unknown predictions, we will go that way for sure. Yeah.
Great, thank you. Yeah, and Sienna had another, starts with a statement that goes into a question. You argued that descriptive DEB parameters can be surrogates for species. Do you think the approach can be extended from species to other biological factors like sex, life stage as well?
So I'm not sure again if I understand correctly. So if we can like add these as more features, is that the question? So like
I read it as instead of just going from species to species, could you go
okay,
from the male to the female based on, some of the data you're getting or different life
yeah,
stages if you had those
yeah.
parameters? That's my read of it.
I'm pretty sure if you would have enough data for that, then you could apply a very similar approach. Not sure where you would find that data, so, but yeah, why not? Yeah,
Might be something to try because often I don't know about as much in eco, but in like human studies using rodents, often it's only the males tested, and then we have to wonder what would happen in the females. So maybe that could be
exactly.
another case where something like this could fill
Yeah,
in some gaps.
yeah, but probably also would make more, yeah, experimental data required for that. Because as you say, for a lot of testing, there's only one sex tested, and I don't know of any databases or collections of data that might make such data available at the moment.
Okay, thank you. Arthur asked, they started with, thank you for the interesting presentation. Have you tried applying causal discovery methods in your work? Considering recent growing interest in these methods like causal/influence diagrams.
To be honest, no, we did not try that. No. Mm. What would you maybe you can add to that? What would you hope to achieve by adding that or replacing some kind of analysis we did by doing that?
Yeah, we'll see if we get a response from
Yeah.
Arthur. Okay, Lily asked, how did you do the semi-random splitting?
Mhm. That's actually pretty easy. So there was a paper using that so it's not our idea, but what they did was just ordering the data set by the toxicity values. Uh, so from from large to small or whatever. And then we just picked every fifth value from that and created our 20% test data set like this. So very easy approach actually.
Great, thank you. Peter asked, what is the distribution of explained variance by the feature set? I may be misunderstanding this, but why reduce to 100% explained variance instead of a lower value like 95%?
Could you repeat that
Sure.
question, please?
What is the distribution of explained variance by the feature set? I may be misunderstanding this, but why reduce 100% explained variance instead of a lower value like 95?
I'm not sure what is meant by that actually. The
That's okay.
distribution of explained variance by all the models or just by one model? I'm not sure. So I can't tell you, we did not assess that yet. Might be interesting if you provide some more information. Again, what would be the added benefit of that?
Yeah, maybe we'll get some more background from Peter. If not, if we can
Yeah.
send an email. Okay, a couple minutes left if that's okay to keep going. There's a few more questions. So one question from John. What data do you think is missing that would improve the overall predictability of your models?
Yeah, that's a very interesting question. We work on that already at the moment. So first of all, it's definitely just more samples. Yeah, so sample size is a big thing. Then we made some restrictions, which I showed you in the beginning. So it's only like one test duration, which we included for that exercise we did here. But there is a lot more data available on time dependent toxicity also, which makes it necessary to use different algorithms, obviously, because we then have to deal with time series data. But we also started on that. So new models we just create can handle that and can also predict toxicity in a time-dependent manner. Yeah, and I mean, there's a lot more biological information out there than just these depth parameters that we tested here. So we are going very broad at the moment, testing different biological traits for their suitability in our models.
Great, thank you. Let's do one last question. You're also getting lots of compliments in the chat as well. So Anna started with a statement, I'm very in favor of the idea of bio-QSAR. I totally agree that for chemical risk assessment, the chemical and biological parts should be combined. If I understand it correctly, this is only for the aquatic system so far. Like the big water solubility problem you have mentioned,
Yeah.
what would you think about the soil environment?
That was also already started. Yeah, I mean, we have one big problem there to start with. That is the common exposure unit in the terrestrial. So a lot of different units are used there to somehow characterize exposure. So that makes it at least a bit more complicated to come up with that. But if that problem is solved, then yeah, I mean, it doesn't matter if you have aquatic or terrestrial organisms. Physiological processes should work pretty much the same, or at least the key processes that yeah, make a species susceptible to chemicals or not. Yeah, so I think yeah, in the future that might also be an extension to come at some point.
Great. Well, we're right at the top of the hour, so I just want to thank you so much for your presentation and answering these questions. And thanks for everybody online for their intention and engaging with the speaker. All right, everybody, have a great rest of your day. Thank you so much.